So how would you choose a topic for your project? This is going to start with those of you that are just planning to, right? You are just thinking about developing a project idea, and maybe you're thinking, how do I do something like a publication that I read? Or maybe you're thinking, how do I do something in an area where I'm naturally interested in, but I really don't have the data to start doing that kind of an experiment? So that's the first point that I think we should start from, is the topic that you already know and care about. So if you're currently doing your PhD, you've selected a topic, you're maybe thinking of a particular experiment, and you want to understand how to make it more impactful. How does omics data factor into this? If you're already conducting experiments and you've collected data, you might be thinking, how do I find additional data that I don't have the capability to generate myself, but I really want to use it to make my project more impactful? Or I want to develop a new hypothesis, but I don't have the means to generate the data. So that would be the first step, is really having a topic that you know or you care about, and to ask yourself, is that a topic that I know and care about? Can you prepare a few slides to explain the topic background? And can you see yourself spending a few months researching the topic? So really, for those of you that are planning some kind of bioinformatics analysis, even though it's just data analysis, it still takes time. So you have to be prepared to dedicate at least several months just to the analysis. The second part is, what will be interesting to your audience? So I'm assuming that the majority of you here are already in some kind of research. And so if you think about your audience, what is that audience? That audience could be your peers, your mentors, other faculty. They could be the broader research community. And this is not a trivial question for any of those audiences. Within a niche, there's already a niche. Within every domain, there are specific topics that are more interesting, they're more on top of everybody's mind. And so that's the second component here. The third component is that you have to have the data, and you have to have some kind of a body of research that is available about that topic, right? So if you are planning to conduct this research and there's no data, you're back at the same place, right? So it's got to be something that comes from availability of data. Now, in between, at the intersection of these three, is the project that you want to focus on. And I want to talk about this a little bit more in detail, because that's where typically most of the participants that we have in our programs fail. They fail to quickly identify a relevant topic that is interesting to a broad community and find data that can help them make some kind of an interesting project out of that question. So I think that defining that question, making it specific, and making it actually possible to execute is a big challenge that needs to be overcome. Now, when people publish, they typically publish from primary data that they have collected, and that is the raw data, the firsthand observation that they have themselves collected. In many cases, you are not going to be the person that generates that primary data, because so much data is already available. But that doesn't mean that you cannot start your project from there. And so what we will do in the context of the first part of our program is we're going to kind of explore all of these sources of data to generate a hypothesis or develop a problem or a question, and then find the solution or the answer. And I think that that has to do with an overview of literature, but also the available data sets. And it has to do with a good founding, a good established understanding of methodology. How have others extracted insights from data sets that they describe? And so the typical process that we recommend is to start from where you are and try to define what is the specific data set, what is the problem statement that you have, and then trying to work through on how to make that into a much more specific question, because that really can help you then narrow down the number of options that you have to do your analysis, and it will streamline or simplify the interpretation at the end. And so the first part is to review available sources and to categorize them by some kind of type. For example, you can think of different types of research articles that present the current topics or that focus on one specific thing. You can focus on a number of different categories, but you have to build out that overview or that kind of a landscape of where you are compared to what's already out there. And then it will be more clear where are the specific gaps, unanswered questions, maybe controversy, right? So some people don't agree with each other, and maybe a debate. And those are typically the most interesting things to address. So that gap, those unanswered questions and those debates around different topics is the place to focus to define your question or hypothesis. So here, what we want to do is we want to kind of review what's out there, make it more specific. Where do we want to focus? And that typically will be in the place of gaps and unanswered questions. Now, a good place to start is from existing projects, right? So a part of review, and by the way, this doesn't have to be just in your domain, is to see how have other people tried to answer questions with data. And so what we've done is over the past several years, we've gone through publications that focus on specific types of problems and specific types of data and have extracted insights from that data by processing, exploring, and analyzing those data sets. So we've collected a number of projects that you can start with as a springboard into the process because they clearly take out the methods, break down those methods into tutorials that you can follow, explain the data set and how the data set was collected and why that data set is an impactful way to study that particular topic. And once you've gone through a few examples, it becomes almost easy for you to start thinking about the data in terms of a hypothesis that could be answered with a given set of tools. And so when it comes to your project, you are really going to be able to take those examples and leverage the same types of methods to apply to a data set of your own. So this is how the program that we envision is going to work. We're going to first follow through and learn about some of the basic data types and how they are processed and analyzed. Then we will introduce case studies, and those case studies will provide you with a good overview of the topics, where the data came from, how it was analyzed, and what are the results. At the end, you should develop a framework of how to apply such thinking to any new data set or a problem that you identify. And so then we will go to focus on your independent project, where you will frame the problem. And framing the problem means writing out the specific question and how you plan to address it. Then we'll go through finding the data sets, and that data will be of different types and different formats that we will review. Process the data for analysis with the right tools, and today I'll show you a few examples. Perform in-depth analysis, trying to develop a statistical model that can either prove or disprove your hypothesis. And then we'll also work with you on communicating the results of the analysis, which is a very fulfilling process because it helps you get feedback early on from a community of like-minded people that will point out all of the details that you need to address for your final project. So let's take an example and think about how to frame the problem. What does that actually mean? So there are several types of problems out there. For example, you can take a clinical problem, right? So we all live in the era of personalized medicine, and we can think of clinical challenges in the context of personalized medicine. For example, patient stratification. How do different patients respond to different pathogens? How do different patients respond to different treatments? Are there specific groups of patients that we should be aware of? Disease classification and diagnostics. How can we differentiate one disease from another? Maybe one disease stage from another disease stage. Maybe it is the predictive component. How can we identify potential responders to therapies that will respond better to a therapy based on omics data, for example? And also we can look at mechanism of disease and disease progression, and that is more of a research question. And nevertheless, it plays a major role in how all of these other questions could be addressed. Another category of problems are related to intervention. And so for the pharmaceutical and the biotechnology companies, what is important for them is to know biomarkers. How can we understand specifically what is the molecular process that can help guide our decision-making, that can help define a diagnostic test? We can think of detection of toxicity, right? When there are interventions, how do we know that those interventions can have side effects? And those side effects could mean toxicity at the maybe in vitro stage. As well as analysis of molecular mechanisms of a drug action or target discovery, right? So how can we use the information that we have about a particular interaction between an intervention and the host at the molecular level and understand what are the targets that we can focus on and how to eliminate the targets that are potentially unreachable or unrealistic. And also a big domain in everywhere, including infectious diseases and non-infectious diseases, is drug repurposing and repositioning, right? So how can we take existing compounds and apply them to a different type of a challenge? Or how can we take the same drug and combine it with another one, or maybe modify its mode of use so that it is positioned in a different way? So after we've framed the problem, we need to find the data. So let's take a look at a few data sources where you can potentially identify a data set for your research, okay? So I'm going to go here to NCBI, and please follow with me to NCBI. This is the link. And please let me know once you've reached NCBI by putting yes into the chat box. And as we're doing that, I want to say that the majority of the people that have answered the first question said number 1. So they're already working on a data of their own. They've collected their data. They're doing some analysis themselves. And 9 people said 2. So 9 people said that they're still planning or thinking about a project. Okay, so please let me know once you've reached NCBI by putting yes in the chat box. Thank you, Mohammed. Encourage. All right, thank you. Okay, it looks like a lot of you have. Okay, so another question for those of you that already reached NCBI. On a scale from 1 to 5, 1 being no experience and 5 being expert, how many of you have experience, or what is your level of experience working with NCBI? So 1 to 5. 5, you're an expert. 1, you don't really know. This is new to you. All right, so I see that Hosea wrote number 5, so I'm going to pass on the microphone to Hosea. He's going to guide us on a brief explanation of how to use NCBI. That's a joke. So for those of you that are very familiar with NCBI, that is a great thing. What I think we want to do today is just look through some examples of data sets that we can quickly find, and I'll show you how you can use them, even if they're very large, even if they're something that you probably don't want to download to your computer and load up your laptop with this data set. So here we have a search box on top that searches for all databases. Okay, so think of any concept that you're interested in or any topic that you're interested in, and let's just see what's out there. So I'm going to take the first 5 examples that you post in the chat. So put down something more or less specific. Okay, so not just the name of the organism or a disease. Propose something a little bit more detailed, a little bit more specific. So what type of a data set, what type of a problem statement, what type of a question do you have that we can try to find data for? Okay, I'll take the first 5 suggestions from the chat box. Okay, any suggestion? Does anyone have any project in mind that they don't mind sharing with the rest of the group? Okay, so whole genome sequence. Again, whole genome sequence is going to be a little bit difficult to define because that could mean anything, right? Whole genome sequence of what? What pathogen? What? Okay, great. So Ahmad, you can try again. Let's go to the next one. Gulshan Tairova, NYC tuberculosis. So I don't know if that's referring to New York City or did you want to say Mycobacterium tuberculosis? But again, that's broad, right? That's very broad. That's just a pathogen. Okay, let's go to the next one. Omatayo is saying drug target for Parkinson's disease. Okay, let's try that. Okay, so drug target Parkinson's disease. That would be number 1. Okay. Abukari is saying microsatellite sequencing data. Again, something I think a little bit broad. Let's be more specific. Applied to what? What kind of a disease or something like that? Okay. Abir is saying RNA microarray data for the LMK gene. Okay. So I have a little bit of a problem with that because if you're using a microarray, you probably don't want to just choose one gene, right? Instead, choose a pathology or choose a specific situation where this type of data will be interested. Let's go on. Jose is saying comparative genomics of COVID-19 strains. Okay, perfect. So COVID-19 strains, we don't even have to search anywhere. We can look over here, SARS-CoV-2 data. So we'll take a look at that example and see what kind of data is out there. Human microbiome and soil transmitted helminths. Okay, Jewelna Akori is asking a very interesting question. So that's going to be number 3. Let's see what we can find for that. NCBI. Personally, I do not know anything about this topic, and that's what's exciting about my job. I get to learn about all of these interesting things from you. Okay, James. So we have 1, 2, 3, 4. So this is the 4th. Okay, so we have here alkaptonuria disease. James, I would recommend being also a little bit more specific. Just the disease itself is probably too broad. So think of a more specific question. Let's go to the next one. Mohammed Sagir Shehu is saying single nucleotide polymorphisms of ACE2. I think it's interesting. So let's take a look. But I would mention that it's probably also very broad, right? So let me, first of all, fix this, ACE2. Okay, so the last one, let's see, who's going to be the lucky person? Plasmodium falciparum data. Okay, so that's interesting, but let's again be more specific. Let's go to the next one. Manzanella species in Africa. Okay, let's try. Okay, so let's go to the first one. So the first project that you proposed was drug target in Parkinson's disease. And so let's take a look briefly at what types of data are available and how much. So there's a lot of books and publications and different kinds of references in literature, which obviously we would expect. But here we have some information about genes. Now, who knows what does GEO stand for? Does anyone know what GEO stands for? Gene expression omnibus. Okay, thank you, Abir. That's in fact correct. So we have here a number of gene expression data. So we don't have a lot, but we have a few data sets. Now let's think whether gene expression is a good way to study drug targets for Parkinson's disease. I think it is a good data type because when we look at how a drug affects a particular tissue or a cell line or a whole organism, what we really want to know is whether there is a protein, a receptor on the surface cell, or some kind of a surface protein that could be accessible for a drug, is playing a major role in this disease. And therefore, if we interact with it, we can potentially make a difference. We can change something. So I think that's a perfect example. So let's take a look at what kind of data do we have here as a drug target for Parkinson's disease. So here we have gene expression of A53T and corrected iPSC driven to DA neurons and exposed to paraquat and Manzanella. Okay, so a lot of gibberish that I personally don't understand. So I would love to learn more. And I can click here for more and read about the organism experiment type summary, the overall design, and potentially even a publication that I could read. But let's go and see what do we actually have? What type of data do we have here? So here it says we have 12 samples. And the important part to think, right, is how can this data inform me about making a reasonable question that I could be answering with such data? So what I see here is that we have 4 different groups. In each group, we have 3 replicates, and those replicates are corresponding with some condition. So if I was to start looking through the data sets, I would need to structure the data in such a way that I can ask questions by, for example, making a contrast between one group and another group, or maybe through classification where I can identify different features that are specific, that are unique to a particular condition. So it looks like if I'm interested in finding a data set to start asking questions about some particular drug or a pathway for a new drug target, this would be a good data set to start. Okay, so for the first one, we found data in about a couple of minutes. Let's go to the second one. So here, it's obvious that we're going to find a lot of data. Every lab in the world that was shut down for any other purpose found a way to repurpose their research to study SARS-CoV-2. And so much data has been generated just in the context of this pandemic that it surpassed every other pandemic before this, including Ebola virus, SARS-1, Zika, etc. In fact, we saw a 4-fold increase in the number of genomes generated every month for SARS-CoV-2 compared to all of the other types of previous outbreaks combined. So I think there's a huge number of data sets. You can see that there are over 185,000 SRA runs. So who knows what SRA stands for? I'm assuming some of you that put number 5 for the previous question are very familiar with these abbreviations. So please let us know, what does SRA stand for? All right, excellent. Thank you, Abir. Sequence read archive, exactly. So here we will find compressed FASTQ files that contain sequencing read data. And from those sequencing reads, we can study gene expression, genome variation. We can identify groups of different viruses inside patients, et cetera, et cetera. So over 185,000 of these, 50 3,000 nucleotide records, so genomes or genes or proteins, clinical trial data, publications, and references. So really a whole collection that you can scroll through. So I'm not going to go in detail. Anything you want to ask about coronavirus, most likely somebody generated some data that you can find and study that. Let's take a look at this one very specific problem here: human microbiome and soil transmitted helminths. So if we look here, we're struggling to find any raw data. We have some publications, we have some books, but we don't have any data sets. So let's try to find, nevertheless, what is going on with these soil transmitted helminths. So the first thing that I already know that there's a lot of data on the human microbiome, so I can pause with that real quick and look for soil transmitted helminths and see if I can find anything for that. And it looks like I can. Now, because I'm interested in the microbiome, I know it's not going to be in the gene expression omnibus data because that's going to be gene expression, and I'm looking for microbiome, a different type of data. So probably I want to go into the bio project and see what can I find here. And here I do have one bio project that deals with the metagenome. Okay, so what do we have here? The impact of enthelmintic treatment on human gut microbiota in Western Kenya, cross-sectional and pre-post deworming comparisons. I think this is exactly the type of data that you're looking for, right? I forgot who asked this question, but do you think this is a data set that could help you study this topic? Okay, whoever, okay, great. So there's your project. So here you have 211 samples already for you, right? You don't have to generate a single sample because first of all, you probably want to understand whether this data set contains some interesting insights into what you are trying to identify. So there we go, a complicated, challenging research question, and we found data for it in just a few minutes. All right, so another question was single nucleotide polymorphisms of ACE2. So whoever asked that, please add a little bit more detail in the chat box so we can kind of take this further, but it's an interesting question. We all have heard that ACE2 is the primary target for the COVID-19 SARS-CoV-2 virus. We know that ACE2 is expressed in the lungs and other tissues, and maybe the question is whether there's a genetic component to people that have acute reaction to SARS-CoV-2 or a milder reaction to SARS-CoV-2, maybe some other types of information like ethnic background or other important information could be a factor here, right? So obviously we're going to find a lot of data here, but I think we need to structure the question better to find good responses. So ACE2, how do we find single nucleotide polymorphisms? Single nucleotide polymorphisms are found in genome sequencing data. And so instead of looking for single nucleotide polymorphisms in ACE2, I would just probably want to find some data set that I could query or I could search through to find polymorphisms typically found in maybe a healthy population or maybe between different tissues or potentially between different species, as I can see here, right? So if I focus on the Homo sapiens ACE2, let's search for that. Okay, here I have, again, bioprojects. So let's take a look at bioprojects. Okay, here we can clear all because we're in the filter for metagenome. And because this is going to be genomic data, unfortunately, I don't see genomic data. I see epigenomics, other, which I don't know what that would be, transcriptome. So it looks like maybe we need to search through. So whoever proposed this, do you have a more specific question here? Okay, I'll wait for you to come back because I think that here, in the context of ACE2, it would be interesting to see what the specific question would be. That does it have polymorphisms? Probably yes. Do you want to just compare different people, or do you want to find a particular position, or how do you want to use that information, right? That's, I think, where you need to think a little bit more and propose how to make it more specific. Okay, so now let's look. We have Manzanella species in Africa. So again, unfortunately, this is kind of broad, right? So I think if we just look for Manzanella, and then we see what kind of information do we have about Manzanella. It looks like we have some geo data, so gene expression data, some bio projects. So let's take a look if any of these are in Africa. Okay, let's see, where was this collected, if we can find out. Looks like a very recent data set. A genomics, okay. Do we know where it came from? Let's see. Um, okay, unfortunately, it does not tell us where it was collected from, and it's a very recent sample. But I can imagine that you would be able to find data depending on what is your research question. What are you trying to find, right? So the point of this is that I think for the majority of the projects that you proposed, so 3 out of 5, we found data very quickly. For the other ones, it took us a little bit of time, primarily because you need to be very specific when you're asking the question, just like those 3 good ones asked, right? It's the microbiome for this particular pathogen, and we found that data, right? Once you can define your question with the type of data, the type of a organism, the type of a specific question that you have, that helps you very quickly answer the question about whether that data exists. And remember, let's go back really quickly to that slide. When you choose your problem, you need to care about it, right? So some of you have proposed your questions, presumably you care about them. Number 2, it has to be interesting to your audience. So that's up to you to decide. But 3, that's an important component. You have to have a data set to start working with to be able to work on that project. So that's an important component here that we always go through. So we've thought about how to frame the problem. Again, several examples of how to frame the problem and how to explore whether that problem could be answered with data. Now let's talk about processing the data. So many of the data sets that we have looked at will require processing of raw data. For example, when we looked at the sequence read archive, those contain individual reads from sequencers. Those need to be pre-processed, then they have to be mapped. They potentially, if it's gene expression, you have to go to quantification, maybe differential gene expression, or you're free to start asking the question, what is the signal to noise ratio? What kind of signal am I looking for that can help answer my question? So in the context of this program, we'll take a look at several types of data sets, including viral sequencing or metagenomic sequencing. We'll take a look at genomic data, how whole genome sequencing, whole exome sequencing, or genome assembly for pathogens works. And we'll also look at transcriptomic data. So we'll learn how to process that data to go from individual reads to a table of expression. A big step that many people overlook is the exploratory data analysis step. So as we saw, we found a number of data sets, and those data sets have been prepared by different labs. different hands did the sequencing. They did it in different conditions. We need to know that the data is solid, that we can use it, that it doesn't have any artifacts that are going to lead us astray. So let's see whether we can find some data set and quickly explore it, okay? So here we have expression profiling by array. I'm just going to use this example to show you what kind of data exploration could be meaningful in this case. Here we have 3 replicates per condition. So let's take a look. How can we explore this data? So here we have the table. So let's download this table really quickly. It's going to be a big table, but we'll take a look at what's inside. Okay, so we're going to take this table and let me unzip it. Okay, I'm just going to open it up in Excel and let me read here a little bit more. What does this data describe? So Parkinson's disease, PD, is characterized by extensive loss of A9 dopaminergic DA neurons in the substantia nigra pars compacta, SNPC. Okay. Okay. A strong association has been reported between PD and exposure to mitochondrial toxins, such as the environmental pesticides, paraquat, manib, and retinone. Here, using a robust patient-derived stem cell model of PD that allows comparison of synuclein mutant cells and isogenic mutation-corrected controls, we identify mitochondrial toxin-induced perturbations specific to A53T syn mutant A9DA neurons. We report a novel molecular pathway whereby basal as well as toxin-induced oxidative and nitrosative stress inhibits the MEF2C PGC1 transcription network in A53T HNS compared to corrected controls, contributing to mitochondrial dysfunction and apoptotic cell death. Our data provide mechanistic insight into gene-environmental interactions in the pathogenesis of PD. Furthermore, using small molecule high throughput screening, we identify the MEF2C PGC1 pathway as a new drug target for therapeutic benefit in PD. So here we know that this is a mutant, right? This is A53T mutant. So A53T, this is the mutant. And here's another A53T, right? So what is PQ? And so I think that these were paraquat, maybe, MB, paraquat, and maneb, and retinone. So maybe these are the pesticides. So maybe this is the mutant with pesticides. This is the control with pesticides. This is the mutant without pesticides. This is the control without the pesticides. So my question would be, as I want to explore this data, would I be able to see clearly differences between the control group and the treated group, or between the mutant and the control? So let's try and figure that out. So here's my data. I'm going to just change this to ID. And actually, I'm going to use this because right now I'm not interested in specific genes. Here, I have the names of probes, it looks like. So okay, so this is not useful. Okay, unfortunately, with this data set, it is designed around probes, and so I cannot just take the data set as a table of expression. What I probably will be able to do is get the raw data and find that data inside. Okay, but theoretically, what I would want to do is I would want to explore this data using something like principal component analysis to see whether the groups separate. And the important next step here is to be able to structure this analysis in depth, right? So here we have an example of what I wanted to show you. Unfortunately, the data wasn't ready for it, which is actually 80% of the case. That's what you're going to find. But once I structure the data and I find the right table, I could perform something like principal component analysis that would help me design the specific contrasts that I could turn into biologically interpretable analysis. Okay, so I'm going to pause here and ask you if you have any questions about what we did and how you are thinking about your own project. So does this ability to find reference data sets on NCBI and public domain repositories, does it help your project? Do you see yourself being able to leverage these resources to be able to ask such questions? Okay, so the other example that I briefly wanted to show you is this one. Okay, so the second problem that we will take a look at today is working with large numbers of data sets. So again, you can see here is there are 211 samples. Okay, so to process these samples, you actually don't have to download the data to your computer. So in the context of our program, we're going to provide you with access to our server platform. That is a big data platform. So please go with me to server.t-bio.info and we'll take a look at the second data set. Okay, so here, what's important is to understand how was this data prepared. Okay, Deepsa, maybe you could share the demo account details. Okay, so what's important here is to understand how this data was prepared. And you can see here that this is amplicon data using Illumina MiSeq, and it is metagenomic. It's PCR and it's paired. And because it is metagenomic using MiSeq, I probably think that this is going to be amplicon data. So what it's going to simplify for us is that it's only going to provide us with sequencing of the 16S ribosomal RNA gene. And that will allow us to study data about microbiome composition and species richness. So if I want to analyze all of these data sets, again, one of my options is to download all of them to my computer and start running a pipeline on my computer, but I can see how this can quickly get out of control because there's just so many samples and each one of them is fairly large. So instead of doing that, what I will do is I will send the results to Run Selector, or over here, go to File, and download the run info table. And what this will do, okay, what this will do is it will create a table from which the server will itself pull the data from NCBI, and I will be able to run this analysis without requiring any kind of computational power from my own computer. So let me show you how to do that. Okay, here we have the runs, 211 runs. And here there's a download link, which typically I could use using SRA tools to download to my computer, but I'm going to skip that. Okay, so here we also have the description. Okay, they were taken from different dates, it looks like. And all of them. Okay, so this table, let's go to the server, could be analyzed with either the data2 pipeline right here or KIND2. So let's use this amplicon 16S example to see what we can learn from this data set. So here we have a demo of what this pipeline would look like. And so I'll just briefly explain. Once the data is loaded, you go through the data2 workflow, which clears up the sequencing reads and prepares a table of operational taxonomic units that is then cleaned up and prepared for analysis using phyloseq that will show us how the distribution of species in different samples varies based on, in this case, I think, infection. So as a result, you will get a number of outputs that include visual outputs and table outputs that could be further used to study these in detail. How do we actually upload the data? Instead of the demo, now with the credentials that BIPSA provided, you cannot do this, but once you join the program, you'll get your own personal account. Here, I have the ability to select Silva as my reference database. I will add the file that I just downloaded as an NCBI run table. So I'll do add file. And here, I'll take this table that I uploaded. And here, I actually don't have a supplementary text file, but I'll just upload something else. And now I can start this pipeline. So without downloading any of the files, just providing the reference links to NCBI and defining what groups those samples would fall in, I can start this pipeline and create for myself a full metagenomic analysis, understanding species composition and richness between those different samples in the microbiome. Okay, so briefly, I think what we went through today is how to find data, which could include looking on NCBI like we did today. It can include raw data on different repositories like SRA, bioprojects, and Geo, and other dedicated databases. For example, somebody mentioned Plasmodium falciparum, so there's a full database of data specifically on Plasmodium falciparum, PlasmoDB. Then we talked about how to do exploratory data analysis, and that can include running something like principal component analysis or looking at summary statistics for my data, understanding whether I can use that data and make reliable inference from my analysis. Then we talked about doing meaningful analysis, translating my question into a model, into a test, in a particular analysis that I will want to run. And finally, you will get to your result. So what is the result? What should be included in the result of my analysis? So as we said in the beginning, you start with the project name and selection of your topic. You then perform specific tasks. You downloaded the files from somewhere, you analyze them in some way. All of those steps need to be recorded. So what's going to be important in the context of this 3-month program is the ability to keep a journal where you record all of this information that later on will be included in your poster, in your report, or in your publication. So there you will describe who generated the data and how. What are the sample names? And how do you find those samples if somebody else wants to reproduce your analysis? What are the specific analyses that you've run? How did you transform the data or manipulate it before you ran an analysis? And so all of those tie in into your results and discussion. How does your result compare to what other people did, especially if you took some other people's data? And what does your analysis contribute? What new information does your analysis contribute to what is already known about this topic?